welcome back to another video, and in this video I will be explaining the information regarding the GenBank. In my previous video I have gone through the overview of the NCBI website, but in this video I will be specifically focusing on the GenBank and the data which is represented in the GenBank. So if I want to access the GenBank, there are different ways by which I can search the information in the GenBank. So the first way by which I'm going to access the information in the GenBank is through the Entrance global search. So for instance, if I want to find the gene sequence of amylase from Bacillus subtilis, what are the steps that could be done? So you can have your own query, but for the reference I'm just going to show a walkthrough using the amylase and Bacillus subtilis. Okay, so firstly what we will do is that in the entrance search bar we will just type Bacillus subtilis. So this is the organism from which we are trying to identify the amylase gene sequence or the amylase RNA sequence. Okay, so we'll put an end as a boolean operator, and here we will write the name amylase, which is an enzyme, and we can simply press on the search button. But whenever we are providing the name of an organism, there is a space in the middle because it is genus and species. Okay, so whenever you represent the name of an organism, make sure to put it into like this, and you can simply press on the search button, and then you can scroll down over here, search, or you identify the place where the genome is written, and within this small box search for the nucleotide. So when you press on this nucleotide, you are directly going to the database, which is a GenBank. Okay, so as you can see over here, all these entries which have come in front of you, there is this GenBank mentioned over here. Now the question might arise in your mind that why there are these other organisms appearing over here. So this is because we have not applied a proper filter. One of the ways is to simply click on the right hand side where Bacillus subtilis is written, or what else you can do is that here in the boolean operator search field, you can simply remove this which says all fields, and we have to be very specific, like we want to identify amylase from this organism which is Bacillus subtilis. So we will remove this or section like this. So now a boolean operator has changed to Bacillus subtilis and in square brackets organism and amylase, and if I press on search, now it will only show me Bacillus subtilis and amylase. So all the entries having this Bacillus subtilis and amylase over here. So you can filter out the way you want. You can play around with these options over here, and you can also play around with the search details and by changing the boolean operators. Now let us just open one of these files which are over here. But before I open this, I just want to show you one thing. That suppose if I just copy this from here, go back to the home page of NCBI, and rather than going through the entrance global search, we can directly select nucleotide from here, and in the search bar we can paste Bacillus subtilis organism, put a boolean operator and amylase all fields, and if you press on the search, you will be redirected to that same page that we had previously seen. So I will press on the search button, and you will come to this place with the same entries over here. Now there is a third way in which you can do the whole process. Like rather than going through the home page of NCBI, in the address bar, after the address which is mentioned at the home page of NCBI, put a slash and write GenBank and press enter. You will be redirected to the page which says GenBank overview, and again all the information about the GenBank is mentioned over here. But for your information, GenBank is a primary database. It has the information only regarding the sequences of DNA or RNA. Now, if I place the same terminologies with the proper Boolean operators, and if I press on the search button, it will again bring me to the same page. Okay, so as you can see over here, there are a lot of sequences of DNA which is mentioned over here. So now let us just focus on each one of these, or just one of these. Now it says that it is Bacillus subtilis, partial gene, AMY gene for amylase. So this is the name of the entry. Now here it says 1469 base pairs. But now when it is mentioned as base pairs, there is a caveat over here. The caveat is that when you open the sequence, we don't know whether we are getting the sequence of the plus strand or the minus strand, because DNA is a double stranded. So the information regarding which strand of DNA is presented in front of us, we don't know, because we don't know what is the 5' end and what is the 3' end in the sequence that we get. But anyways, I will explain you step by step what I am trying to tell. So yeah, so this is the entry that we want to first open. It belongs to this GenBank. And I will explain what is FASTA, what is graphics. But first of all, let us just open this sequence for now. So as soon as I open this sequence, the data entry in the GenBank looks something like this. Okay, it is a simple text information which is represented in front of you. Why? Because it is a primary database. And the most important thing in this entire page is this accession ID. This is again very important. So like if I know this accession ID and I copy this, if I go back to the home page, okay, I choose a nucleotide over here, and rather than putting the whole field over here, if I know the accession ID, I can directly paste it over here, and if I search it, it will directly take me to that place where that data is placed. Okay, so now this number that you see, this ID that you see, is actually called as a accession ID. Now what does this mean? So like when you purchase a vehicle, each vehicle has its own number at the front and at the back of the car, or two-wheeler, whatever you have. So each vehicle has a unique ID. Similarly, each entry in the GenBank is given an ID, and it generally appears over here like this. Okay, now as you can carefully see, there is a full stop and there is one number over here. So this means that this is the first version, the original version of the data which is represented, and this data is not been modified. If this data is modified in future, or if any data is modified, then the number after this full stop, after this dot, will change. Yeah, so if you are working with some kind of a data set with multiple sequences, you need to remember this ID, or you need to keep this ID somewhere within the notepad file or within the CSV file, or whatever your data might contain. Okay, so for this entry that you see, this is the unique identification number which is given to this entry. So now let us look into the architecture of the data which is there located over here. So the first thing that you see over here is a locus, and the locus has the same accession ID which you see over here, but the number or the version number is not mentioned over here. Okay, version number is mentioned over here in the topmost column, but it is not mentioned in the text file over here. If you want to specifically search for the number of the version, you have to look in for this place which says version, and it says 0.1, means it is the original data which was submitted. So when I say data was submitted, when it was submitted over here, or it was published on this date. So 26 July 2016, this data came into GenBank and it was publicly open for the researchers to get an access. Then the next thing that you see over here is what is the length of the sequence. So it says 1469 base pairs. Okay, it belongs to the DNA, it does not belong to RNA. Even if it is a RNA, I will there are a few caveats I will tell you later. It is a linear sequence, it is not a circular sequence, it is a linear sequence which has been deposited over here. What is the title of the entire entry? The same title which you see over here is mentioned under the definition. So definition means the title of the entire sequence. Accession ID, it is the same as the locus. Version, I have already mentioned. What are the keywords? The keywords is amylase, and it is AMY gene, and source of the organism is Bacillus. Again, the organism is Bacillus, and this is the entire family, genus, taxa, or the taxonomic information is represented under this organism. Okay. Then the next information that you get is reference. There is a single reference. Who are the authors, like who have submitted the sequence to the GenBank? So these people, and these are the authors, or these are the researchers who have submitted the sequence to the GenBank, and it got published online on 26 July 2016. So that is what we are trying to get. Now, what was the original title when they were doing research? So the original title was Cloning and Sequencing of the Gene Encoding Amylase from Bacillus subtilis. This is a strain number isolated from cultivated soil in Vietnam. So this is the full information about the entire entry that you see, which is under the title. And what you get on the top over here, whereas what you get under the definition is the sort of a running title or the short title of the entire sequence. Then it says that this information has been published in any specific research article or not. So it says that this is unpublished. So this data is not published in any research article. Okay. Then here it says authors. Okay, so it is the same authors or who have provided this information. It is the first author in this case. Then the next thing that you see is the title. Now the title means that who has submitted the data. So this data is directly submitted by this author, and so the title says direct submission. Okay, and then it says journal, and it is submitted. So it is not accepted. So this information is not accepted anywhere, but the information which was there was originally submitted to the journal for publication, and the information is mentioned over here. Okay, so these are the basic information about the authors, about the bacteria, about the gene, about the name of the gene, about the date where it is published, accession ID, and stuff like that. Then the next phase of the data set is basically called as the feature, and in pure terminology it is the starting from this point where features are written up to the point where this ends somewhere over here. So this highlighted section or the selected section is actually called as a feature table. So it may not appear to you as a table, but in the actual context it is the table. This information is in the form of a table, and it follows certain rules. So the first information is source, and it says that the sequence starts from the position number 1 and it goes up to the end 1469. So there is no missing information within this region. So as you can see, 1469, it is the same 1469 base pairs we are talking about. So it starts from the first position and ends at 1469 position, and then it belongs to Bacillus subtilis. Molecular type is a genomic DNA. Isolate means the strain number that they are talking about. From where does this organism was isolated? So this organism was isolated from the soil. Then what is the taxonomic information about this organism? So this number is mentioned. So if you click on this number, it will take you to the place where you can get the taxonomic data of this organism. Then what is the location? So the location is Vietnam. Now within this region, from where does the actual gene starts? Okay, so first 75 nucleotides is just the upstream region, and from the 76th nucleotide the actual gene starts. So the actual gene starts at position number 76 and it ends at 1469, and this region between 76 to 1469 is actually called as AMI. Okay, the same CDS region means which part of the DNA is giving rise to mRNA, which is giving rise to a protein sequence. So that coding region starts from position number 76 to 1649. It says gene is AMI, codon number 1. So here what does it mean? It is trying to tell that which codon it starts. So there are reading frame, so it starts with the first reading frame. Okay, so there are 3 reading frames in the given sequence. Okay, so out of this 3, it starts with the first reading frame, and other protein information about this enzyme, it is mentioned in this part of the section. Now when it is translated, so when this DNA sequence is mentioned over here, so when this DNA becomes mRNA and then that mRNA becomes a protein, the protein sequence of this gene looks something like this, where each alphabet in capital letters means that amino acid. So it is a 1-letter abbreviation of that amino acid sequence which makes this enzyme called amylase. Okay, so this information starting from this point to the ending of this amino acid sequence of the protein is actually called as the feature table. Okay, now this is a very important part. So this is the actual sequence of the data which is deposited into this gene bank. Okay, so now as you can see over here, each alphabet is 1 nucleotide. Now here we don't know whether this sequence is of a positive strand or a negative strand, which part is 3' and which part is 5'. Okay, so we have to look into that consideration later when we are dealing with the sequence. And each alphabet is ATGC because it is a DNA sequence, so it is ATGC, and in the small alphabets. Okay, now as you can see, this sequence is in the form of a cluster. So this is one cluster of 10 nucleotides, this is again 10 nucleotides, this is again 10 nucleotides. So it starts with position number 1. So this is the 10th nucleotide, this is the 20th nucleotide, 30th, 40th, 50th, and this is the 60th nucleotide, because there are 6 clusters each of 10 nucleotides. So the first line consists of 60 nucleotides. So the next 61st nucleotide is over here, which is a T, and its number is 61. Okay, so why is this arrangement so important and why the data is arranged in this way is because suppose if someone tells you that find the 101st nucleotide, so you can quickly go like this. So this is 70th, 80th, 90th, 100th. So this T is a 100th nucleotide, and then this C is a 101st nucleotide. So you can easily count visually by looking at the sequence if it is represented in the form of a cluster. Okay, so this is the first nucleotide and this is the final nucleotide. Okay, so what is this final nucleotide? This final nucleotide is 1469. So this is 1469. So if you count, this is 1, and if you keep counting up to the end, it will be 1469. Okay, so this is how the entire sequence is located. Now what else do we come to know from this? It says the actual gene starts from 76th position. Okay, so actual gene starts from 76th position. So this is 60th, so the 61st, 71st, okay, and here it says 76th, 71, 72, 73, 74, 75, and 76. So this is the actual coding region of the DNA, and the sequence which is above this blue highlighted region is the upstream element of the gene. Okay, and when this part is translated to become a protein, it will give this sequence which is mentioned over here on the top. Okay, so this is very important, that you need to know from where does your gene actually starts. It does not start from number 1, it actually starts from this position which says 76th to 1469. So 76th from 1469. Okay, and at the end of the entire data there are 2 slashes which represents that the entire data ends at this position. So the ending of the entire data is represented with this 2 slash. So this is the outline of the data which is there on the GenBank. Now there are 2 crucial parts over here, one which says FASTA and the second which says graphics. Now the FASTA is the representation of this sequence in a particular format. Okay, so when I click on this, so just remember, so the first 3 nucleotide is AAG. Now if I just click on this number which says, or this link which says FASTA, it gives me the sequence in this way which says AAG. It is the same sequence, but here what you see is that the sequence, the alphabets have become all capitals or all caps. The clusters are not in a form of group of 10, it is a continuous sequence. Okay, and it starts from position number 1 and it ends at whatever the position number that was. Okay, now this, starting from this greater than symbol to this point where it ends, this entire part is actually called as a FASTA format. It is not called as a FASTA file, mind well. It is called as a FASTA format. So what are the basic features of the FASTA format which is there for any sequence from the GenBank? Okay, so the feature goes like this. So it is a greater than symbol, then followed by the accession ID, followed by the organism, then it says partial gene, partial AMI gene for amylase. So this is actually this entire line followed by this greater than symbol is actually called as the address line of the FASTA file. Okay, then you hit enter, go to the next line and you directly paste the sequence as it is. So this becomes the entire sequence in the FASTA format. Okay, so this is the FASTA format, it is not the FASTA file. You can copy this sequence, put it into a notepad file or any text editor, and then you can convert this sequence into a FASTA format file. But this is just a FASTA format, it is not a file. Okay, so whenever you see this greater than symbol followed by the address line and then the sequence starting from the next line, it is actually called as a FASTA format. So whenever you see this kind of arrangement to any sequence, it may be a protein sequence, it may be a DNA sequence, it will always start with a greater than symbol, address line followed by the entire sequence. Okay, so this was the basic idea about the FASTA file, sorry, not the FASTA file, the FASTA format and the data which is there in the GenBank. So I will just go back and I will tell you a small caveat about the GenBank. Now whenever you open any data into a GenBank, even if it is a RNA sequence or if it is a DNA sequence, you will never find uracil, because RNA sequence generally will have uracil. So even if you open any sequence from the GenBank, it will never have a uracil. Why it will not have a uracil? Because when you sequence an RNA, the RNA is first converted into cDNA, which is the reverse transcriptase influenced product of the mRNA. So when reverse transcriptase enzyme is converting the mRNA sequence or any RNA sequence into a cDNA, it becomes the DNA where uracils are replaced, and the sequence which is a DNA, which is an RNA sequence, converts into a DNA sequence, which is then sequenced. Okay, so even when, if it mentions in any of this, say if you find any other any other GenBank file, and if it is mentioned mRNA over here, but if you scroll down, you will never find a uracil. Why? Because that mRNA was then converted into cDNA with the help of reverse transcriptase enzyme, and then that mRNA become the cDNA. cDNA does not have a uracil. Okay, and then that cDNA is sequenced using Sanger sequencing or any other kind of sequencing, and then it is deposited into the GenBank. Okay, so once again, a short recap: you can access GenBank directly through selecting nucleotide, or directly going through the GenBank, or you can use the entrance search to go to this place and come to this place. You can use Boolean operators, and you can use advanced search. So if you are not familiar with the Boolean operators, what else you can do is you can go to this advanced. Here you can search for say for example what you want to search. So here you can choose an organism which says Bacillus subtilis, and here you can choose all fields, and you can write amylase, and then you can search. You will come back to the same place, because automatically this Boolean operators and this search tools or this search format will be automatically given by the advanced search tool within the nucleotide database, which is the GenBank database. Again, there are a lot of filters that you can choose from. Accession ID is absolutely important, and the version of the accession ID is again very important. So this is all what I wanted to tell about the GenBank data structure and the FASTA format of the sequence. Thank you.